Welcome to Shay's Microbiology. Now that we have wrapped up bacteriology, this is where things start to change a bit. Up till now, with bacteria, we have been very comfortable. We could see them, we could culture them, we could stain them, identify them, and test them against antibiotics. Viruses don't give us that luxury. In laboratory virology, the first thing you have to understand is this: viruses are not cells. They don't grow on routine media, they don't multiply on agar plates, and they don't exist independently the way bacteria do. A virus cell only makes sense inside a living host. So when we talk about laboratory diagnosis in virology, we are no longer asking what organism can I culture. We are asking what evidence of viral presence can I detect. And that shift is very important. In virology, the lab is not chasing the virus itself most of the time. The lab is chasing signals, viral antigens, viral nucleic acids, or the host immune response to infection. That's why laboratory virology relies heavily on indirect methods. Now, historically, viruses were studied using cell cultures, embryonated eggs, and laboratory animals. And yes, those methods still exist, especially in reference laboratories. But in modern diagnostic laboratories, especially clinical labs, the focus has moved strongly towards serology and molecular techniques. So instead of growing the virus, we detect viral DNA or RNA, viral proteins, or viral-specific antibodies in blood. And this is where virology starts to overlap with immunology and molecular biology in a very serious way. Another key thing to understand is that timing matters in virology. The stage of infection determines whether the laboratory can detect. Early infection, you are more likely to detect viral antigens or nucleic acids. Later infection, antibodies become more useful. As we move forward, we'll talk about how viruses are classified, how samples are collected and handled, and the major laboratory methods used to diagnose viral infections. But for now, the foundation is simple. Viruses change how the laboratory thinks, how the laboratory tests, and how results are interpreted.